All right, these are going to be your notes on Punnett squares, why we use Punnett squares, and how to use Punnett squares to predict probabilities of offspring traits based on parent alleles. So the first thing we need to know is that Punnett squares are a tool used to determine the probability that a certain allele will be passed from parent to child. Remember that you can pause, speed up, or slow down this video however you want. So if I go too fast, just pause and wait, because I'll definitely be going too fast because I'm going to kind of cruise through these. So Punnett squares are a tool used to determine the probability that a certain allele will be passed from parent to child. Some alleles are dominant, and some are recessive. Dominant just meaning that that is the trait that will be shown. Recessive is the trait that will be masked if there is a dominant allele. Dominant alleles use a capital letter. So if the gene you're looking at is height, individuals can either be tall or short. And so we note whether individuals with that gene have a tall allele or a short allele with these letters. Capital T equals tall. Lowercase t equals short, because recessive alleles use the lowercase of the dominant allele. So if the dominant allele is tall, we use a capital T. That means that short is the recessive allele, and we use a lowercase t. You must always use the same letter when we're working with these Punnett squares if you're talking about a particular gene. So, if an individual has at least one dominant allele in their genotype, genotypes are the letters we'll use in the Punnett square signifying their genes, then their phenotype, how they physically look, so PH for phenotype, PH for physically look, genotype is for genes. Then if they have at least one dominant allele, they will show that dominant trait. So example would be, if you have two dominant alleles, you are tall. If you have a dominant allele and a recessive allele, you are still tall. Only if you have a pair of recessive alleles would you be short. A person with two dominant alleles is said to be homozygous dominant. Homo meaning the same, zygous referencing their genes. So they have their two genes that they have are the same. Hetero means different, zygous referring to their genes again. So if they have one dominant allele and one recessive allele, they are heterozygous. Sorry that watermark is getting in there. And then if they have two recessive alleles, they are homozygous recessive. All right, now we're going to work a couple Punnett square problems to show you how this works out in practice. So here's how a problem would look. It would say cross a, we'll call it homozygous dominant dad with a homozygous recessive mom. So big T dad with little t mom. We separate dad's genes and put one above each column. We separate mom's genes and put one beside each row. Dad's genes drop down into any box that is below them. Mom's genes move across into any box that is beside them. And so this is telling you the probability of their children, their offspring. This is telling us, and we'll talk about this in terms of genotypic ratio and phenotypic ratio, that the genotypic ratio means that 4 out of 4, or 100% of their kids, are going to be heterozygous. They're going to have one dominant allele and one recessive allele. Phenotypically, 4 out of 4, or 100% of their kids, are going to be tall. So that's genotypic ratios and phenotypic ratios. Genotypic means what are their genes going to look like? They will have one dominant allele and one recessive allele. Phenotype means what will they physically look like? They will all be tall. because so long as you have at least one dominant allele, you are going to manifest that dominant allele. So they have one big T, so they are going to be tall. All right, one more example, and then you'll be ready to work some problems on your own. Let's say we cross two heterozygous individuals. So big T little t dad and a big T little t mom. So we make our Punnett square. Dad's genes get separated, one above each column. Mom's genes get separated, one beside each row. Dad's genes drop down. Mom's genes move across. This is more of like a grammatical thing. The dominant allele always goes ahead of the recessive allele, so I could have just as easily written this little t, big T, but that's just grammatically incorrect. It still gets the same idea across. You just always put the dominant allele first. So in this case, our genotypic and phenotypic ratios are going to look a little different. 1 out of 4, or 25%, will be big T, so homozygous dominant. 2 out of 4, or 50%, will be big T, little t, heterozygous. And 1 out of 4, or 25%, will be little t, little t. But the phenotypes are going to look a little different because this person has at least one, or these people have at least one dominant allele, so they are going to be tall. So actually, 3 out of 4 of these children are going to be tall. 1 out of 4 is going to be short. Remember that these are just percentages, so if these people have 4 children, it isn't a guarantee that 3 will be tall and 1 will be short. All 4 could be short. These probabilities only work out over hundreds of thousands of permutations. So if these people had 1 million children, you could pretty safely say about 750,000 would be tall and about 200,000 would be short. All right, those are the notes on Punnett squares. Talk to me if you have any questions.